Spermatogenesis. So this is what? Sperm creation, right? So we're going to talk about sperm creation, how it's formed, where it happens. So we said, where does sperm production take place? It takes place in the testes, right? So we have these special type of primitive cells. We call these the stem cells of the reproductive system. In the males, these are called spermatogonia. Spermatogonia. These are the stem cells of the male reproductive system. And the uniqueness about them, every time they go through some kind of division, what happens is, because they go through a process here, as you remember before, that we talked about with division, is mitosis, they generate a exact replica of the original cell, and they also create another one that goes back in the specific location, right? And then we talked about chromosome number, why it's important. So we abbreviated N for 23 chromosomes. So if you ever see 2N, which is what a spermatogonium has, this is saying it has a 46 chromosomes. And what was 46 chromosomes? Was it haploid or diploid? These are diploid, right? And so after the first division of mitosis, so you remain in the 2N state, now we have the primary spermatocyte. So primary spermatocyte is also a diploid, right? And this diploid here goes into this next division here, so we'll keep everything symmetrical, through the first phase of meiosis. So now, spermatogonium to spermatocyte is all mitosis, so everything is an exact replica. However, now we have genetic variation when we go through the process of meiosis. So the first step of meiosis, so Meiosis One, takes place where the primary spermatocyte becomes a secondary spermatocyte. So the first phase of meiosis in terms of separation, in terms of division, is identical to mitosis, right, in terms of number. So the number of chromosomes still remain. So now you have secondary spermatocytes here, right? And you need to go through the final step of division, right? So you have mitosis, Meiosis One, and then the final step of division here, which is referred to as Meiosis Two. So in Meiosis Two, because after the early division, every time you have a division in mitosis, you have a sense of DNA proliferation, right? There's an instance where that happens. In meiosis, it does not happen. You have a replication with no sense of a replication with no time for the DNA to begin to reduplicate, right? And so because of this phenomenon of meiosis 2, now this diploid becomes a haploid. It has half of this genetic material that we talked about, so it has only 23. And what is this called? This is called spermatids. So spermatids are immature sperm cells, right? And they're all produced in the testes. However, these spermatids need to become mature. So then they begin to migrate into where? Where does the maturation of sperm happen? As you remember, it's the epididymis. So the epididymis is where we have the maturation of sperm. So in the epididymis here, now you have these spermatids that are still haploid that become more mature. They develop the specific tail, everything. They get the acrosome, and they develop the neck in order for the storage of ATP in mitochondria, right? And now they're known as sperm cells here. Now we have a sperm cell. And this is basically the process of spermatogenesis, start to finish.